Well, hello. This is now lecture 13. One thing is you can congratulate yourself. You've gotten through this far in the course, and I hope that you've learned a lot of great information that can be applicable to your healthcare profession, as well as any other circumstances you might find yourself in. We are going to be dealing with nervous system infections and wound infections in this last lecture. I do strongly encourage you to review the key terms and the glimpse of history at the very beginning, since many of these points will be useful, as well as many of the terms will be useful. Now, again, this course presumes that you've already taken Anatomy and Physiology 1 and 2, so you have some familiarity with the nervous system. And so we're going to start off with the nervous system. And with that, we're going to move forward here. Now, motor nerves are nerves that carry messages away from the central nervous system, brain and spinal cord. Sensory nerves carry messages toward the central nervous system. Thus, many viruses are using axon transport, where they enter via the sensory nerves and they migrate to the central nervous system. Now, we've said this is composed of the brain and the spinal cord. You have to keep in mind that the CNS is going to be enclosed by bone and bathed by a cerebral spinal fluid, CSF. As a result, the CSF may also be an indicator of the presence of a viral infection. I mean, as a nervous system infection, it could be viral, it could be bacterial. Axons are thin, long extensions which allow the transmission of action potentials. Many viruses will be transported through the axonal cytoplasmic transport system. And when we talk about ganglia, these are actually clusters of cell neuron bodies. Keep in mind that, of course, the cell body, which will include the nucleus, may be quite a distance from a target tissue, and its communication means is an axon, whereby action potentials will travel down the length of it. Now we have to also get familiarized with some other terms that involve infections of one type or another. Encephalitis is a generalized infection or inflammation of the brain. Remember that the brain, and to a lesser extent the spinal cord, are protected by these meninges. There are three of them: the dura, arachnoid, and pia. You also want to keep in mind that in the arachnoid space and the arachnoid web structure provides spacing for blood vessels and capillaries, and also will have cerebral spinal fluid passing through it. Meningitis is an inflammation or infection of the meninges. If one has meningioencephalitis, this is actually an infection of both the meninges and the brain. And finally, we have to keep mindful of the fact that the pathways to the nervous system are not straightforward for the bloodstream. This is because of the blood-brain barrier, which is a selective barrier composed of the capillary epithelium, the basal lamina of that capillary, and the astrocyte pseudopodia. So the bloodstream is a very good transport medium for pathogens, can deliver pathogens next to the brain, but actual exposure to brain tissue requires that the pathogen breach the BBB, blood-brain barrier. Many viruses can do this. It is believed also that prions can do so as well. Infections close to the brain, such as above the mouth, may involve veins that transport blood to the brain, may involve veins that transport blood to the brain, or actual arteries. So you have a conduit of pathogens to the brain. Now, nerves, you're going to deal with another issue here. This is very particular with tetanus toxin, herpes simplex virus, and rabies virus. Many pathogens will travel from the synapse, where they made a connection, to the cell body via what is referred to as retrograde, that is, reverse transport up the neuron axon. Others will travel anterior grade, forward, transport from the neuron to the neuron via the synapse. Now, in the bone, we have spaces in the bone, sinuses, mastoids, middle ear, that may provide sites of pathogen culture. then transport into the brain. Also, skull fractures can be a means for entry of pathogens into the brain. That has to be kept in mind. Finally, we have to deal with microglia. Now, microglia are macrophage-like cells. They may actually allow viruses to reside in them. One case is for the HIV virus. Current evidence suggests that although nerve cells do not become infected with HIV, supportive cells within the brain, such as astrocytes and microglia, as well as monocytes and macrophages that may have migrated to the brain, can become infected with the virus. Researchers postulate that infection of these cells can cause a disruption in the normal neurological functions by altering cytokine levels, by delivering aberrant signals, and by causing the release of toxic products in the brain. What am I saying here? Certain unique cells in the brain allow for viruses to, in some cases, stay hidden or to hide there and cultivate in such a way to cause damage to the actual biochemistry of the brain or the eventual behavioral outcomes from the brain tissue. Another thing that's important to note is that, and this was something that was going on with HIV and infection research, was that you didn't always have the delivery to the brain of the specific antiviral drugs. They couldn't be effectively going across that BBB. But the virus, well, the virus was there. And that's why a lot of times there was big concerns when you did have a patient go into remission. They were afraid, well, the virus that's lurking around in astrocytes, microglia, or monocytes or macrophages in the brain might eventually be the reservoir that would lead to a resurgence in the viral count later on in time. Normal microbiota. There is none. Since the brain and the spinal cord are surrounded by tissue, this is usually a very sterile environment. And I do encourage you to review microassessment number 1 for this particular chapter. First off, and here's a nice area of the brain. I just wanted you to take a peek at this. Keep in mind that in the spinal cord, there is a space down in the lumbar region where you can obtain cerebrospinal fluid. This is extremely important because cerebrospinal fluid can be a very good indicator of whether there is a neurological infection, whether it's meninges or actual nervous tissue. But you have to understand this. Sometimes these viruses and sometimes the bacteria are not going to be as easy to detect, and CSF will usually indicate this at a point where, in some cases, the infection has gone much further down the road and led to subsequent neurological damage that will not relieve itself even if they do completely cure the patient of the infection. As for bacterial infections, what we're going to be talking about first and foremost is meningitis. And what you see here is Streptococcus Pneumoniae. And this is, of course, a gram-positive diplococcus. And what you see is an image that from concentrated CSF of a person with pneumococcal meningitis. I would turn your attention to table 26.3. And what you're going to see is a very clear comparison of the different agents that are involved in causing meningitis. Now, meningitis in and of itself can be marked by an accumulation of white blood cells in the cerebrospinal fluid, swelling in the brain. Almost all bacteria can be found there. Haemophilus influenzae is usually a type that you're going to find in young until a vaccine. Now it's very present in cases of meningitis for older adults. Streptococcus algae, this is where babies acquire this from their mother's genital tract at the time of birth. Streptococcus pneumoniae mostly affects adults, usually preceded by otitis media, sinusitis, and pneumonia from the organism. And what you can do is see this very nice histogram here of the breakdown of the various types of organisms that will cause the meningitis, and looking at the age groupings and the percentage of the cases. So you see there's a much greater amount of group B streptococcus at less than 1 month, and this shifts, and you see a much greater amount of Streptococcus pneumoniae as you get older in the population, post-adolescence and over 60 years of age. There is a sort of middle peak that occurs between 2 and 18 years for Neisseria meningitis. So moving forward here, what you're going to see here, this is a meningococcal type of arrangement, and you have polymorphic nuclear neutrophils here. And what you have here is the petechiae from meningococcal disease. Remember that petechiae are basically small breaks of blood vessels. They look like purplish spots, and it's caused by bleeding underneath the skin. So let me move forward from pneumococcal meningitis, from streptococcal pneumoniae, and then we'll go through the other types, and we're going to eventually get to that table that is extremely helpful. Here it is right here. For pneumococcal meningitis, this is the leading cause of meningitis in adults. Begins with a mild cold, sudden onset of throbbing headache, fever, pain, stiffness of neck and back, nausea, vomiting, confusion, coma, and death may occur due to the inflammation and the shock. Streptococcus pneumoniae is a gram-positive diplococcus. Serotypes include strains with protection from phagocytosis via the polysaccharide capsule. The common cause for otitis media, the inflammation of the middle ear, sinusitis, and pneumonia. This can enter the bloodstream and then pass into the cerebrospinal fluid via attachment to meningeal cells and causing meningitis. Damage is mostly due to inflammation. But you have to keep in mind it can cause intracranial pressure and death. The epidemiology is usually you're going to have bacteremia, most found is in this type of infection, and the case fatality rate is less than 10% in young adults, but higher in the elderly. Treatment is penicillin, and for antibiotic-resistant strains, use of ceftriaxone. Also, you may include vancomycin. There is a treatment, there is a preventative in the sense of a vaccine. Now, meningococcal meningitis, well, what you've got there, humans are the only host. The symptoms are very similar to pneumococcal meningitis, as the symptoms will appear in 1 to 7 days. They include petechiae, though, mild cold, followed by headache, fever, pain, stiff neck and back, and vomiting. The petechiae, which I showed you earlier here, the breakage of small capillaries. They look like small, tiny bruises or purplish spots on the skin. And this is due to the bacterial outer membrane fragments released into the circulation. These fragments are endotoxins and trigger an inflammatory response. They will result in a drop of blood pressure, vasodilation, and capillary leakage, and hence capillary damage. This is important to keep in mind that this can result in septic shock and death as a result. The causative agent is Neisseria meningitis, a meningococcal gram-negative diplococcus. The bacteria adhere by pili, entering the upper respiratory tract, entering then the bloodstream, carried to the meninges and spinal fluid. Inflammation will lead to blocked cerebrospinal fluid, and this is going to start a cascade, causing an increase in CSF pressure and impaired brain function. This can usually lead to infarcts and thus death of the brain. Also, you can have the release of the endotoxin, and this can lead to damage of motor nerves that can result in paralysis. Now, how do we have this spread? This is by airborne droplets, crowding, fatigue, predispose one to the disease. It is very common in the dry season in the African, went a little ahead there. There we are. The African meningitis belt. These are the nations there that you see have a high risk of it particularly during the dry season. Prevention and treatment, a polysaccharide vaccine does exist. Rifampin is given to those who have been exposed. Penicillin in high dosage or ceftriaxone for treatment. And as I said, a vaccine is available. Haemophilus influenzae meningitis, this is the leading cause of meningitis for children under the age of 5. Very similar to other types of meningitis for the symptoms. Older children report a stiff neck. Infants may show bulging fontanelle. If you remember now, if the pressure is building up and you have this very flexible top of the head, such as with newborn infants, the fontanelles may begin to rise up. The disease may progress rapidly to coma and death. Treatment with ceftriaxone is the recommendation. Rifampin, prophylactic upon exposure to HIV-infected velvets. HIB, as in the Haemophilus influenzae, okay? Not HIV as in Victor, the virus. So, HIB, of course, would be talking about Haemophilus influenzae bacteria-infected patients, okay? Now, there is a nice case presentation on 26.1. I encourage you to review that. By now, you're getting used to taking on the case presentations or case studies for some of your weekly assignments. Moving forward, and you see this happy little soul there. The rate of serious Haemophilus influenzae diseases per 100,000 children, less than age 5. If you'll notice, in the process of about 20 years, we have really reduced the risk or incidence of this. And this was because of the availability of a conjugate vaccine in late 1987. This influenzae type B was the most common cause of bacterial meningitis in preschool children. And of course, the vaccine now is readily given to little children. Okay. We have neonatal meningitis. And this is difficult to tell from a neonate on a headache or stiff neck. They don't just sit there. They may scream and yell and be unhappy, but usually the signs that are much more providing with information are going to be fever, vomiting, irritability, poor feeding, lethargy, and again, bulging fontanelles because you're going to have increased intracranial pressure. Now, this is a little bit different. The causative agent is commonly caused by Streptococcus algalatia, and this is in newborns. It's a basic bacteria that's found in the vagina of the mothers. Also, it's referred to as group B Streptococcus, so it's called GBS. Now, you will get some cases occurring from encapsulated strains of E. coli, and they came from the mother's intestinal tract. Other cases are caused by Listerio Monocytogenes, and that came from the mother's infected blood. Bacteremia in the newborn leads to inflammation that leads to increased intracranial pressure that will block the CSF flow and result in hydrocephalus. This will result in brain damage. You will also get reports of decreased blood flow to the brain and brain abscesses. To help you understand, keep in mind the brain is now contained in a skull. Even if you have the flexible cartilaginous tissues of where the fontanelles are, you have an increase in pressure. It's going to cause increased risk of trauma to the brain, and that may lead to eventually also brain abscesses. This can lead to deaths in 6 to 20% of the infected newborns. The treatment and prevention is intravenous delivery of ampicillin and gentamicin mixture. It should also be important to test the women late in their pregnancy for GBS in the vagina and rectum and treat with the proper antibacterial medication. Next we're going to go through is listeriosis. Oh, and by the way, one of the interesting things that they found, list shield. This is a phage preparation. Remember phages? These were particular phages that could block, or they were particular to destroy different types of bacteria. In this case, the list shield is sprayed on foods ready to eat. And the phages will not harm the humans, but what they will do is destroy any Listeria bacteria present. And since meningitis can be a manifestation of listeriosis, that's why I'm including it at this point. What you're going to have, though, is fever, muscle aches with or without gastrointestinal symptoms. You will have also headache and stiff neck, which mark the onset of meningitis. Infected pregnant women can deliver prematurely or miscarry, sadly. Listerio Monocytogenes, a non-spore forming gram-positive bacteria, is the cause, usually from ingestion of Listerio Monocytogenes. This is an infection in the intestine and enters the bloodstream. Later, the infection will arrive into the meninges and cause meningitis. How do we get this? This is usually by contaminated dairy products, soft cheeses, coleslaw, hot dogs, non-pasteurized milk. Pregnant women can get the infection, leading to bacteremia, fetal infection, and miscarriage. Immunocompromised patients are at high risk because they won't be able to fight it off as well, and it can lead to, yes, even death. Care in handling and cooking raw meats, reheating cold cuts and hot dogs is the prevention strategies. The treatment is penicillin. And a bacterial phage food additive kills the bacteria on the foods, as I've talked with you about before. The next area we're going to move into is Hansen's disease, otherwise known as leprosy. This is anesthetic skin lesions. Anesthetic meaning they're not feeling any pain. The individual will have a deformed face, loss of fingers and toes. Now, if you got to understand this, what's going on is it's caused by the Mycobacterium lepridae, which is an acid-fast rod bacteria, but it's because it favors cooler parts of the body, which are the extremities, ears, nose, fingers, toes, okay? The bacteria will invade small nerves, the sensory nerves of the skin. Activated immune cells will attack and kill the nerve cells, producing nerve damage. As the cells favor cooler parts of the body, such as skin macrophages and peripheral nerves, this form is called lepromatous leprosy. The tissues and mucous membranes have billions of bacteria cells, but no inflammatory response. But the mucus of nose and throat contains cells, making it easily transmitted. Okay? Tuberculoid leprosy, this is where the cellular immunity limits the growth of the bacteria. It's not transmissible in this state, but immune system eventually gets overwhelmed. There is, for the epidemiology, direct contact with M. lepridae from the mucous membranes. It's been interesting because it was found to exist in armadillos and specific select species of monkeys. You can see how the fingers are being affected by this person's hand. Pigmentation cells have already been destroyed. And so what's happening is the cooler parts of the finger are where the bacteria is going to proliferate very, very well. Okay. There is no vaccine. There is no treatment. It used to be, sadly, only isolation for these individuals. Now we have a treatment of dapsone plus rampapin for months or years. Clofamizine has been added for lepromatous disease. Now we're going to move into botulism. Now, before I do, by the way, this is Mycobacterium lepridae. Remember, this is an acid-fast means of detection, so you want to keep that in mind. And you can see the individual bacteria here. And this, the armadillo, is a natural reservoir for the M. lepridae bacteria. So let's start talking about botulism. Notice the endospores, that's that bulging there, okay? It is caused not by the bacteria itself, but it is caused by the toxin made by the bacteria. Symptoms include blurred or double vision, weakness, nausea, vomiting, diarrhea, generalized paralysis, and respiratory insufficiency. You see, what's going on is the toxin is acting on the respiratory neuromuscular junctions, and it blocks the acetylcholine neurotransmitter release. The bacteria, Clostridium botulinum, is an anaerobic endospore germinating organism, bacteria in food, and it will release a neurotoxin. You can see the endospore bearing cells as well as the vegetative cells here. And how you get the release of the neurotoxin? Well, the neurotoxin can survive enzymes in the stomach acid, and it will be transported by the bloodstream to motor neurons and initiate paralysis. C. botulinum can also colonize the intestine or wounds. In warfare of, let's say, 100 years ago or 200 years ago, it was very common for C. botulinum to be the reason that there were tremendously high deaths. There are several images out there in the 1800s of soldiers. They took one picture of an individual. He was barked on his back, etc., and he was suffering from botulism poisoning caused by the wounds from his war. And it was not some of the other bacteria that were killing him. What he was being killed was the botulism from the battlefield. This can be in the soil. It'll also be in some of the field. The best way to put it is you'll have dead animals, manure. You know, if you had basically cavalry riding through the area, they'll have cuts and wounds and everything else, and so they get these infections. A lot of times they died from these infected wounds than they would have died from actually the bullets or the sword. Also, botulism has been used as a biological warfare agent. Keep in mind it's the neurotoxin that does all the action. And the endospores are present in the soil. The toxin binds irreversibly to the presynaptic membranes of the peripheral nervous system, particularly neuromuscular and autonomic nerve junctions. The toxin binding blocks acetylcholine release, and this is going to result in a weakness, a flaccid paralysis, and often a respiratory arrest. The cure occurs following sprouting of new nerve terminals, but a lot of times what happens is the individuals die before that could ever occur. The spores are widespread in soil, aquatic sediments, dust, etc. And what you see here is actually food, and the cases where you will have this due to, and you can see the comparison, foodborne botulism, infant botulism, wound-based botulism. Okay. So what do? Well, first off, you have to have proper canning of produce. That's why in the canning industry, their D rating for canning and heating and preparation goes to a level of 17. Treatment, enemas and stomach washing to remove the toxin is another approach. Cleaning infected wounds of dirt and dead tissue, and the use of an antitoxin, and if necessary, artificial respiration. And the artificial respiration is to keep the person breathing until eventually the toxins are purged out of their system and are no longer shutting down the release of acetylcholine. Now, we're going to start focusing in on viral diseases of the nervous system. Now, I want to bring this up. You need to be aware of this, that, many different viruses can also affect the nervous system, including Epstein-Barr virus from such things as infectious mononucleosis, mumps, rubiola, varicella zoster, herpes simplex, and there's also certain zoonoses. Usually the number of infections are low and the incidence is rare, but we have to talk about a couple of these that are very important. First off, viral meningitis. This is also referred to as aseptic meningitis. Symptoms: abrupt onset, fever, stiff neck, severe headache, vomiting, sore throat, large parotid glands, rash, and chest pain. This is usually caused by a Coxsackie virus, which will cause the throat and chest pain, or an echovirus, which will cause the rash. Both of these are RNA enteroviruses of the picornar virus family. Mumps is also common in unimmunized populations. Okay. The viremia from the primary infection site seeds the meninges. There's no decrease in CSF glucose. There's fewer leukocytes that enter the CSF than in the bacterial infections. The epidemiology is usually fecal-oral route of transmission. The enterovirus, you see these type of outbreaks mainly summer, early fall. If we're talking about mumps, this is going to occur fall and winter. Prevention and treatment, hand washing, avoiding crowded pools during an enterovirus outbreak, and of course, regularly taking the mumps vaccine to be resistant to mumps. Okay. The next we're going to be dealing with is the viral encephalitis grouping. Now, if you take a look, this is West Nile virus. But there are a bunch of arboviruses that cause encephalitis in the United States. Now, an arbovirus is by definition a virus transmitted by some type of arthropod. An arthropod is a general classification for things like insects, so that could include fleas and mosquitoes, but it can also include, in some cases, ticks, okay? Because those are part of the arthropod family there. Let's talk about this. So the symptoms are fever, headache, vomiting, one or more nervous system abnormalities, disorientation, localized paralysis, deafness, and coma. Sometimes sporadic viral outbreaks of measles, mumps, infectious mononucleosis. Mostly this is caused by arbovirus, arthropod-borne viruses. These are all enveloped, single-stranded RNAs. The lacrosse encephalitis virus is a Bunya virus family. St. Louis and West Nile virus is a flavivirus. And the Eastern and Western encephalitis virus, these are toga viruses. Okay? So you can see that. Let's move over. And what you see here is the incidence of West Nile virus infection as of 2011. What you have to understand that West Nile was sort of detected in the late 90s and spread westward. It first started showing up on Long Island and a couple of areas in New England, and then eventually spread by the mosquitoes that were passing it along. Okay. Now, the replication is at the site of the mosquito bite. Further replication will occur for the virus in the lymph nodes. Viremia will eventually seep or sneak into the brain tissue. The nerve cells are invaded and destroyed. Now, the virus is transmitted to humans from birds or squirrels via mosquitoes. That's why for a while they would say, if you see a dead bird, notify so-and-so, because what happens is dead birds, usually West Nile will kill the bird, and they've frequently found like dead crows or dead birds or whatever, and they're just dead on the street and overwhelmed by the virus. That's why if you see in the notes, they'll talk about chicken sentinels. These are basically birds that if they get sick, you'll be able to see it, and therefore you know that the virus is out there, okay? Also, what we really have to do is go after the main vector, which of course is the mosquito, and that's where you bring in insecticides. At present, there is no antiviral treatments for these type of viral encephalitis viruses. Animals are used to monitor the existence of the pathogen. The animal must be able to be infected by the pathogen, with or without the help of a vector such as the mosquito. Now, you see the distribution? Let's move to something far more threatening, and that is polio. Formerly also referred to as infantile paralysis, poliomyelitis. The symptoms will include stiff neck, headache, fever, nausea, pain, muscle spasm followed by paralysis. Now, This is all due to a poliovirus, which is a member of the Picorni virus family. The strains are 1, 2, 3. The virus will infect the throat and intestine, circulate via the bloodstream, and enter the motor nerves of the brain and spinal cord. The infected cells will lyse to release new virions. Now, what you see up here is the result of people infected with polio. This is, yes, one whole ward of polio patients. Because what happens is they get this type of paralysis that occurs while the viruses are entering the motor nerves and destroying the capability of these people to do some of the basic things. Remember, one of the basic things that we see with skeletal muscle is the diaphragm area expanding and contracting, and thereby allowing us to breathe. And what you see are a variety of iron lung machines. These are individuals who have been attacked by polio. They're trying to fight it off. In the meantime, though, they have to be assisted because they've lost the capability to breathe on their own. The intercostal muscles, both internal and external, as well as the diaphragm, have been temporarily put out of action. So without any ventilation, you have no breathing, you have death. Okay? Now, you'll see here, this is some evidence of polio infection because of the type of gait that occurs in the feet. The interesting thing is there are two vaccines, Salk inactivated vaccine, the Sabin oral attenuated vaccine. When the Salk and Sabins came out, you saw the need for treatments crash, basically decrease as the number of cases decrease. So did the vaccine work? You betcha. But let's go back to this artificial ventilation for respiratory patients and physical therapy and rehabilitation. These individuals are going to need help to regain the strength and capability to use the muscles for their limbs and other parts of their body, where because they had been put out of action for a while by the virus infecting and blocking the neurotransmitter release, the muscles will eventually atrophy, and so they have to be brought back up to full use. Okay? Now, there are a couple of other things to be aware of when it comes to polio. There was a situation that occurred in the 1980s for a while, and it was called Post-Polio Syndrome. It was caused by the early death of nerve fibers that sprouted to take on the added functions of muscles that had lost their original innervation due to the polioviral demise. Now, these overloaded nerve collaterals began to start degenerating 15, 20 years after the original illness with the symptoms. So the patient started reporting again muscle pain, weakness, and muscle degeneration. It looked almost like a polio attack. And for a while, there were a lot of people scared that maybe polio had come back or had come back in a way that it was resistant to a vaccine, etc. That was further from the fact than you could ever believe. What they eventually found was that some of these collaterals eventually started dying out. And so that's why the person needed further physical therapy, etc., to attempt to continue to restore some lost function. And perhaps even as the activity continued, they would sprout basically more collaterals and lead to resumption of the muscle function. Epidemiologically, I want to bring up one other area, and that is the effects of the onset of vaccination. Now, the virus can spread via the fecal oral route. It can be asymptomatic and non-paralytic in some cases. OPV, which stands for basically the Sabin oral attenuated vaccine, and IPV is the Salk inactivated vaccine. And what they found is that OPV will provide herd immunity and mucosal immunity to prevent wild type of virus from spreading to the environment, such as sewage. The IPV prevents paralytic poliomyelitis, but not the spread of wild-type virus in the environment. Okay, what was found by the Israelis, and it's mentioned in the book. Okay, I encourage you to take some reading of this. It is on page 711, and I believe there is a little bit more detail of this. If you go to the end of the chapter, and no, I stand corrected there, but there is some mention of this. What happened was that one particular country, Israel, was using one type of virus, but they found that they were still having wild type of poliovirus show up in the sewage. Now, for a while, they couldn't get anything but OPV, and they started having these increases in the virus showing up in such places as wastewater, sewage, etc. And they didn't want to have something where it would come back later on and affect people. So they called in all the people who were vaccinated, but were not vaccinated with the oral vaccine. It was only the IPV that allowed this shedding of wild virus. And what they did was they gave them the other type of vaccine too, to make sure that they would be protected completely. Okay? Now, the next one we're going to talk about is rabies. Of course, the symptoms are fever, headache, nausea, vomiting, sore throat, cough at onset, later spasms of muscles and the mouth start locking down. This is where you get the lockdown stage. Later spasm to throat, coma, and death. Causative agent is a rabies virus, single-stranded RNA, a rhabdovirus as it's called. It is enveloped and it has an inner structure that has a bullet shape. And you can see this on this image here, okay? Now, the principal means to basically transfer the rabies is by a bite to humans in the saliva of a rabid animal. The virus will multiply at the bite site, travel via the nerves to the central nervous system. The virus infects brain tissue to cause encephalitis, and infected cells exhibit what they call Negri bodies. Okay, the virus spreads outward via multiple tissues to infect the heart and other organs, including the salivary glands. This is where you get into the point of the salivating rabid animal. We'll also get into the eyes and into fatty tissue. By getting into the eyes, a patient will start showing off what's called photophobia. They will literally, it's painful for them to see normal amounts of, let's say, classroom light or something like that. And so they have to have it very, very darkened. Okay? Inhalation can also be a positive route. And then you have the pets and the animal immunization. This is one of the ways for prevention. Let me explain to you that transmission can occur via organ transplants, such as cornea transplants, contacts with dogs, bats, raccoons, skunks that have it. That's why we get all our happy little dogs vaccinated, and you can't get a license for the dog for the town without the vaccination evidence. One other thing to consider is that after someone has been bitten, post-exposure washing of the wound with soap, water, and antiseptic is a possible means to prevent an infection. Use of rabies vaccine or an anti-sera. And then also, once the symptoms begin, there is no really effective treatment. Sad to say, individuals, once they start showing rabies, they're down the hill. They're going down the hill fast. You just need to make them comfortable, okay, prior to their death. Okay, I do encourage you to take some time and go over the perspective and the micro assessment. Now, oh yes, and there's our little stinker, the little skunk. By the way, in Massachusetts, just to help you to have some perspective, every once in a while, either via skunks or raccoons, they would have outbreaks of rabies. And since these animals can get interactive with more domestic animals like dogs and cats and things, that's where they would take helicopters and go over certain areas and drop pellets of food, but the food also had in it the vaccine, with the idea that you would prevent these animals from being carriers of the disease down the road. And you would also be able to neutralize some of those cases that might exist in the animals. At least it was an attempt. Fungal diseases. Well, let's talk about this. Now, fungi rarely infect a healthy people. They are very common in the soil and in decaying vegetation. Infection depends on the immune status of the patient. Once infections occur in the brain, due in part to the blood-brain barrier, antifungal medication is very difficult to penetrate into the brain to stop the fungal infection. Now, cryptococcal meningitis. This is an uncommon disease until the onset of AIDS. And now it's very common in AIDS patients. You have headache, vomiting, confusion, weight loss, slight or no fever. Symptoms may progress to seizures, paralysis, coma, and death. Causative agent, Cryptococcus Neoformans. This is an encapsulated yeast. It is also an emerging disease. Cryptococcus gotti is found in the northwest and the western United States and Canada. Now, here's the deal. Cryptococcus gotti can be found in healthy patients. Cryptococcus neoformans is an opportunistic pathogen in immunocompromised patients. The infection starts in the lungs. Its encapsulated organisms multiply, enter the bloodstream, and are transported to other parts of the body. Eventually, the meningitis and brain are infected. Okay. Now, you notice around here you got capsules. You can see this because this is a negative stain. You see in the deep center, there's the cells. There's also the capsules. More virulent strains of C. gotti raise the fatality rate from 5 to 25%. And this also causes a type of fungal pneumonia. Now, these fungal organisms are worldwide in distribution in soil and vegetation. Infection occurs by inhaling contaminated dust. Person-to-person transmission does not occur. Unfortunately, there's no effective means for prevention, and the treatment is amphotericin B with fluconazine or itraconazole. This may require, though, an intrathecal delivery system, which basically is a drug delivery tube that is put into the brain vesicles, because the antifungal drugs do not cross the blood-brain barrier. Okay. Here is another image here, and you can see the halo-like structure, and that's the capsules that prevent macrophages, etc., from being able to consume them and destroy them. Okay. Now, that being said, you can see also the spread of Cryptococcus gotti in the Pacific Northwest in 2008. And you can see how it's spreading out, particularly around the Vancouver Island area, but then it's starting to go down into the coastline, down to Oregon. Okay. The next we're going to deal with are protozoan species of the nervous system. A few species are very important. First, well, we got to talk about a term here. Parasitemia is the level of parasites in the circulating blood. And so we have African sleeping sickness, otherwise known as African trypanosomiasis. And the symptoms are that you have a tender nodule at the site of the tsetse biflight. The vector that transfers this particular protozoan is the tsetse fly. You have fever, enlargement of lymph nodes, later involvement of the CNS, uncontrollable sleepiness, headache, unsteadiness, coma, and death. The sickness, though, may persist for years. Now, let's see. You can see here's another picture of the trypanosomas. There are two major types, Trypanosoma brunsii, and in the Trypanosoma brunsii, there's two subspecies, Trypanosoma brunsii gambesian and rhodesian. Okay? The rhodesian creates a disease much more aggressive and tougher to treat. Here is the nasty little animal called the tsetse fly, Glossinia. Okay? So this is the vector. Excuse me. The protozoan multiplies at the site of the insect bite, later enters the blood and lymphatic circulation. New parasites are released, and they evade the immune system by antigen shifts due to surface antigen changes to surface glycoproteins. The body's immune system is constantly trying to keep up but cannot. Rodentia is a rapid action protozoan that will attack the heart and brain within 6 weeks after initial infection, whereas Gambesian, its progress is going to be much slower. Involvement of the CNS leads to slowed responsiveness, indifference to feeding, sleepiness, coma, death. Now, here's the other problem. The wild animal reservoir for T. brunsi rodensis is why they're having such a difficulty trying to control this. Bites of infected tsetse fly will transmit trypanosomes via infected fly saliva. What are the prevention? Protective clothing, insecticides, pentamine prevents Gambesian form from occurring for several months. Treatment is Suramim, or if the CNS is involved, Malarsaprol or ethronethine. Can also, it's very common, and that's why it's important to ask your patients, where have you been? Oh, we went through the entire African subcontinent as tourists. Well, that's probably why they got the disease. Now, the next one, toxoplasmosis. Now, toxoplasmosis, you'll see this image, figure 2620. Okay, this is very serious. Many healthy patients are asymptomatic for this particular protozoal parasite. Others exhibit sore throat, fever, enlarged lymph nodes and spleen. The symptoms may subside over weeks or months. Now, here's the problem, though. Immunodeficient people, reactivation of a latent infection can lead to encephalitis manifested by confusion, weakness, seizures, stiff neck, paralysis, and coma. Photosensitivity is also common. Fetal toxoplasmosis is acquired by first-time exposure to mother to a protozoan during pregnancy. This can result eventually in miscarriages or stillbirth. Babies born will have seizures, intellectual disabilities, retinitis, and epilepsy. Now, the causative agent, Toxoplasma gondii, is an obligate intracellular protozoan. It is an apicomplexan. It's common in domestic cats and is passed as an oocyte in the cat feces, which is why there was such a problem about that mother in the previous slide, changing the litter box for the cats. Also, this organism will emerge after consumption as a sporozoite and spreads to heart, brain, muscle via blood and lymphatics. Consumption of undercooked meat with mature oocytes is a possibility. And evidence exists that T. gondii alters brain chemistry by increasing dopamine levels. One of the interesting things is that this particular parasite, which is found, interestingly enough, in cats, has been found to release a particular enzyme that boosts the amount of dopamine present. And there's some people that have actually theorized, although all the data is not in yet, that this may account for why some individuals have schizophrenia, where they have lots of cats, and it's not that they brought it upon themselves, but by changing litter boxes, etc., and being exposed, they eventually got enough of the toxoplasmosis in them that eventually it elevated the levels of dopamine to the point where they are perceived as insane or unstable. Okay? Epidemiologically, this is worldwide infections, and it's due to handling undercooked meat or raw meat with tissue cysts or gardening, handling soil in areas with stray cats. Soil and vegetables have feces containing contaminated cysts. What you really got to do for prevention and treatment is this. Cook all the meat. Watch carefully all your fruits and vegetables. Pregnant women should avoid cat litter boxes. Patients treated with pymethrine, which blocks folate metabolism, which is necessary for DNA replication. in combination with the sulfdiazine drug, the sulfa drug, you're going to have some very good progress. But immunosuppressed patients that test positive for an antibody to T. gondii are presumed to have a latent infection and must be given prophylactically cotrimoxazole. But here's the problem: the treatment doesn't destroy any tissue cysts. Okay. From here, we're going to move to the primary amoebic meningioencephalitis, and this is caused by another protozoan called Naegleria fowleri. Okay. Symptoms: fever, headache, nausea, vomiting, stiff neck, coma. This Naegleria fowleri, as well as another amoeba, which is Balthuma mandrilis, an acanthamoeba species, may cause the disease. Now, usually what happens is this. You're going to hear this coming up this summer. Listen carefully in the news. Oh, several children died. Or this child died by swimming in this very warm water in a pond down south. The amoeba must be present in the water and in large numbers. Water containing the amoeba must get up the nose. And once there, the amoeba moves up the nasal passages to the brain. So basically, it will crawl up through the cribriform plate, get into the brain, and it's going to multiply, causing the symptoms associated with the disease. Death will occur within 3 to 10 days following the infection with the organism. Naegleria fowleri is distributed widely around the world. It's present in soil and in virtually all natural surface waters with lakes, ponds, and rivers. Warm water temperatures caused by hot summer months allow the amoebae to multiply because of their food source, the bacteria, increases during hot weather. Thus, the risk may be greater in very warm water, particularly shallow waters. PAM cannot be transmitted from person to person. So you can't transmit primary amoebic meningioencephalitis from person to person, but it is very, very present in some of the warm water lakes and ponds and swimming holes that kids go to. And of course, if they dive in head first, they're going to get the water pushed up the nose, and then you have a problem. There has not been any real successful treatment of PAM. They try using amphotericin B. They may also add ramphopin, tetracycline, and minocycline. This is one of the few free-living protozoans that's pathogenic to human beings. I encourage you again to review the microassessment. Now from there, and here you can see Acanthamoeba, its trophozoite and mature cyst, as well as Naegleria fowleri. And you actually see here the signs where they're warning people, stay away, keep your head above the water to prevent amoebic meningitis. Okay. Now let's talk about transmissible spongiform encephalopathy, TSE. It's a prion-based pathogen. It includes mad cow, which is basically contaminated meat that goes from the cattle, bovine to human. Scrapie, which is a disease that is also prion-based in sheep. Chronic degenerative brain wasting, or chronic degenerative wasting disease, that you find in elk and deer. Kuru, passed from human to human, and Creutzfeldt-Jakob disease. Now, what are the symptoms? Behavioral changes, anxiety, insomnia, fatigue, muscle jerks. Disease basically will progress over months, and you end up with a lack of coordination and dementia. Now, the prions-based marin characteristics, they increase in quantity during the incubation of the disease. They resist inactivation by ultraviolet and ionizing radiation, as well as formaldehyde and heat. They're not readily destroyed by proteases, not destroyed by nucleases. They're smaller than the smallest virus. And they are composed of basically just a protein coded by a normal cellular gene. Excuse me for a second, I had to take a little drink of water. But here's the deal. They're modified after transcription. Prions are improperly twisted improperly in tertiary shape proteins, they will start to replicate in dendritic cells where they convert normal proteins to abnormal proteins. Once these are transmitted to the brain, the prion conversion leads to the aggregation of prions in neurons, and this eventually leads to the death of the neurons. The cell deaths occur, and eventually you see this spongy appearance of the brain tissue slices. Now, human-to-human transmission via corneal transplantation or by contaminated instruments has been demonstrated. Possible transmission of cattle prions to humans via the meat has been transmitted. So it does have the capability of jumping species barrier. Contaminated sheep has been found to infect cattle. And how this happened was in Britain in the later part of the 1980s. This is partly dependent on how Europe versus the United States gives protein supplements to their cattle. Here in the United States, we provide protein supplements by adding grains such as corn or soybeans. They don't have those rich protein supplements available in such bulk quantities. So what they started to do was they started to do what's called animal offal. In other words, tissue remains of slaughtered animals, dry it, grind it up into protein supplement, and feed it to cattle. Problem: they were taking sheep that basically had scrapie, a prion-based disease. Now, scrapie is where they start having the neurodegeneration of the brain. As a result, they start taking on self-destructive behaviors. They start rubbing up and down, scraping their bodies against posts, trees, etc., till they literally wear off the wool off their body and eventually start digging into and having open wounds. Well, the farmers recognized as a problem. The sheep were slaughtered. They were reduced to feed meal, and the problem was they helped spread this into the food, basically the food chain, because the cattle were then slaughtered and used for meat for humans. Okay? So, how do we prevent this? How do we treat it? At present, there is no treatment. It is invariably fatal. Inactivating the prions by autoclaving with a 1 normal sodium hydroxide, in essence, very, very concentrated solution, can best be done, but the best form of tissue disposal is fire and basically incinerate the tissues. Now, with that, I encourage you to review the microassessment, future opportunities, disease and review, and the chapter summary. From here, we're going to jump across and get into wound infections. And this is going to be extremely important that you take some time to review the key terms as well as the glimpse of history. Let's start talking about some of this here. Here is a nasty wound, and if you look very carefully, you'll see that there was some stitching there, and it was popped open. Now, all wounds may be contaminated by microorganisms in the air, skin. The object causing the wound, whether the microorganism started a disease, depends on the virulence of the organism, the number of organisms entering the wound, the status of the host immune defenses, as well as the nature of the wound. Was it a puncture, a crushing wound? Whether any foreign matter or dirt was added into the wound. Wounds with foreign matter usually become infected and will not heal until the foreign matter has been removed. Okay? This is, yes, including things like bullets. Okay? Foreign matter may also provide surfaces for biofilm development, as well as affecting the site of the wound. You decrease the available oxygen, which allows for the growth of anaerobic organisms. Wounds are classified by the following: incised, cut as with a knife or scalpel in surgery. Puncture, penetration by a sharp, small but sharp object. Nail, pin, rake tine. Lacerated, the tissue is torn. Contused, blow that crushes the tissue. And then finally burns. This is serious as the tissue may appear dead and require debridement. Now, thermal burns, although initially are sterile, becomes septic as the microorganisms will grow on the weeping of the tissue burns. If you went over this in the integument in AP1, you note that as if you have a third-degree burn and you've got it now looking as blackened or gray or white, crispy critters, you have to debride that. In other words, remove that tissue, scrape it off. Once you have the exposed burn site, you're going to be losing tissue fluid, and that's going to create the necessary rich medium to grow in bacteria, but so will also the burned up tissue. Of more than 100,000 hospitalized burns, 12,000 die from the burns each year. Now, if we look at the anatomy and the physiology of wounds, we're going to get into, and this is in figure 23.1, the process of wound repair. You've got to go over and review the steps. You got to keep in mind that there are proteins. Upon the wound damage, various proteins are exposed that are the binding sites for pathogens. These include collagen, fibronectin, fibrin, fibrinogen. Now, the first onset is this granulation tissue, which is nodular red translucent material that is in the beginning stages of wound healing. Pyogenic granulomas. Pyogenic, remember, pus producing. This is a wound that contains dirt of the infection that is covered up by granulation tissue. Okay? Due to the infectious material, this wound may not completely heal properly and will result in abscess formation. That's why we always try to clean the wounds. Debridement is the term for the medical procedure to remove dead, damaged, or infected tissue. Debridement helps wounds to heal faster. The other issue, though, is wound abscesses, and I refer you to figure 23.1. Localized collection of pus is present. This may contain both living and dead leukocytes. You will have tissue breakdown products, as well as infecting organisms. Also, as the abscess grows, the pressure by the accumulation of pus causes it to expand. This expansion will occur in the direction of least resistance, okay? Thus, the antibiotic, and basically you're going to get a rupture, a discharge outside of the body. Now, if you have no blood vessels you're not going to have any blood vessels in the abscesses. And so the abscess, which contains, toxins, dead tissue, living cells, dead cells, etc., you're going to have a problem here trying to apply antibiotic into that abscess because it's only going to be dependent upon diffusion, which is a slow process. Generally, abscesses must burst to the body's surface or be drained surgically in order to effect a cure. And as you can see here, you have a wound that has some taping over it. But the problem is you start seeing the infection. If you look on the far right, you see already the formation of pus, okay, which is causing the separation of the two sides of the wound, okay? Now, anaerobic wounds. Clostridium tetani favors growth in anaerobic wounds. Anaerobic conditions are favored in dirty wounds, wounds with crushed tissue. Remember, crushed tissue means what? You're not going to have a blood supply effectively going through there, okay? Because basically the circulatory system has been inhibited due to the crushing effect. Also, you will get anaerobic conditions in puncture wounds. Bullets and other projectiles can carry fragments of skin or cloth, this contaminated with microbes, into the tissues. Also, due to the force by which they may enter, projectiles usually cause tissue damage in the pathway into the tissue. So what I'm saying there is you're going to have this bullet going through and damaging tissue as it continues to travel through the tissue. Let me give you one other insight. I was reading a paper recently where there was a woman that kept shooting up. Now, obviously, an injection into muscle of something like, heroin or some type of opiates. She kept using a dirty needle. Now, instead of getting HIV, what they found she was starting to get was Clostridium botulism, because the botulism were being deposited, the botulinum bacteria were being deposited deep into the muscle, away from oxygenation, away from the surface, and they started to develop in this release botulism. The individual, even though she was partly recovered, the doctors insisted that she stay, and she left, presumably to go shoot up again. But she died when she had gotten home from this. They pumped into her anti-sera for the botulism and everything else. And as soon as she could find a way to get out, she got out. Okay? So the main point I'm trying to say is certain injuries, if they're not properly cleaned, will become massive growth factories for various anaerobic organisms. Okay? Next, we're going to deal with is actually bacterial wound infections. The possible consequences of wound infections include delayed healing, formation of abscesses, extension of the bacteria or their products, particularly the toxins, into adjacent tissues or the bloodstream. You want to keep in mind that infections can originate at an implant site, such as an artificial hip, as the bacteria will presently, if they're on that implant, they're going to develop the biofilm on it. And that is going to require the removal of the implant as part of the infection control strategy. Also, infected surgical wounds can split open, causing the stitches to pull through the tissue, weakened by the infection. Okay? All right. Now, I want you to take a look at table 23.1. We have a very nice comparison there of the leading causes of wound infections. Staphylococcus aureus, Streptococcus Pyogenes, and Pseudomonas Aeruginosa. Let's start with Staphylococcus wound infections. You got to keep in mind that Staphylococci are pyogenic. They produce pus. They're going to also lead to swelling, redness, and pain. If the infection has spread systemically, the fever is also going to be prominent. The strains usually are going to produce toxins, usually superantigens, that can lead to toxic shock fever, high fever, muscle aches, life-threatening shock. The causative agent is gram-positive cocci of the Staphylococcus aureus or Staphylococcus epidermis. Now, Staphylococcus aureus will produce collagenase, which reacts with prothrombin to convert fibrinogen to fibrin. This will then allow the bacteria to be coated with fibrin and thereby resist phagocytosis. The bacteria may also produce or possess clumping factor, which aids in the colonization of the wound surfaces as the bacteria clump together. I would refer you back to table 22.3 on 577, page 577, for a list of all the virulence factors. They also have to keep in mind that Staph A can also lyse red blood cells to help supply nutrients for the bacteria. As we go to Staphylococcus epidermis, this is common cause of small abscesses around stitches used in surgery. They can also colonize catheters and other implantable devices, forming a film. The biofilm protects the bacterial cells from phagocytosis. Here's Staphylococcus aureus under a transmission electron microscope. And here you see the bacteria with leukocytes. And this is the table 23.2 for its review of Staph A and Staphylococcus epidermis infections. The pathology, of course, with Staph A is it can form biofilms. Protein A can contribute to intense inflammation, production of pus. Staphylococcus toxins include superantigens, thereby leading to toxic shock. Staphylococcus epidermis, the organisms can come loose from the biofilms and become transported through the bloodstream. This can result in subacute bacterial endocarditis or multiple tissue abscesses. The epidemiology is interesting because these types are present on either nasal carriers or from the skin. So what are you going to do? You got to first and foremost remove the dirt and dead tissue from an injury, a wound. Treat with an anti-staphylococcal medication immediately after surgery. There are several challenges here. The onset of MRSA, methicillin-resistant Staphylococcus aureus, exists, as does VRSA, vancomycin-resistant Staphylococcus aureus. And this is all due to the R plasmids they may possess. Now there is a new drug that's been approved by the FDA, Synergistic, and that is used, as is also linezolid, and that's a Zyvox for its trade name. Okay. Also, take time to review table 23.3. That's where we're going to get into the necrotizing fasciitis. And this is the flesh-eating disease due to Streptococcus Pyogenes. Okay. And what you see here is also an individual with Streptococcus Pyogenes flesh-eating disease. Okay. So what's going on here? Go to 23.3 and let's go over this. These do include invasive infections leading to pneumonia, meningitis, puerperal, that is childbirth fever, as well as streptococcal toxic shock. At the site of the surgical wound or trauma, or even without an injury, you can have the severe pain, swelling, fever, and confusion. Overlaying skin becomes tense at the site. It may become discolored due to swelling and later shock and death. Streptococcus Pyogenes, the beta hemolytic gram-positive chain-forming cocci, are involved. More virulent strains have the superantigen pyogenic exotoxin A, as well as the protease, which breaks down the tissue, exotoxin B. Now, the pyogenes have fibronectin-binding protein that aids in the colonization. The fascia, which is the fibrous tissue underneath the skin, have been destroyed, and the muscle tissue may eventually be destroyed as well. Bacteria in the dead tissue produce toxins while using the tissue breakdown products as nutrients. The toxins and the organisms enter the bloodstream. The superantigens can cause shock, which is a release of the cytokines. Epidemiologically, history demonstrates the presence of this for centuries. Underlying causes that increase the risk: diabetes, HIV, in other words, AIDS, for immunosuppression, cancer, recent surgery, alcoholism, abortions. This is usually the self-induced or the back alley abortions that occur. Childbirth, and even some individuals with minor cuts. Due to the rapid spread of the disease, amputation is sometimes necessary. By the way, amputation was the standard treatment for World War I. Got a bullet hole, got a beginning of an infection, the only way to save the patient is lop off the arm, the leg, etc. In Civil War era, 1860s, almost 100%, or pretty much 100% of their patients would basically have infections. Many procedures were basically standard. They come in with a battle wound, and let's say it's on a limb, hand, a leg, foot. You chop it off. That's it. And hopefully they'll survive through the infection, etc., and live. Okay. Penicillin can kill the bacteria, but you got to keep in mind this: not in dead tissue areas. That's why a lot of times they would have to then go in, debride the dead tissue, because you're not going to get transport of antibiotics into dead tissue. Dead tissue is not going to have active bloodstream delivery, okay? And the antibiotic does not affect the activity of any toxins that are produced, okay? So let's move on to the next one, and that is Pseudomonas Aeruginosa infections. Now, you notice the green at the end of the limb there that they've had these treatments on. You'll notice the midsection where there is a plate there with the green, and then you notice the bottom part, which is where you see the singular polar flagellum of the Pseudomonas. This is an opportunistic pathogen that is widespread in the environment. It is a common nosocomial infection and occurs in lung biofilms in cystic fibrosis patients. They can release a pigment. The yellowish is pyoverdin, the blue pigment is cyanin, and that results combined in a greenish color in wounds and in growth media. Usually you'll find this in burns or other wounds, and the greenish pigment will actually be present in the wound. The bacteria can be transported in the blood. Then you get symptoms which include fever, chills, skin lesions, shock. Now, Pseudomonas aeruginosa has a single polar flagellum. The pathology is that it induces tissue damage, prevents healing, increases the risk of septic shock caused by circulating bacteria cells, not the toxins in this case. Some strains include toxin A, which halts protein synthesis in host cells. There are other strains that will produce an unusual enzyme called phospholipase C. Now look, if you remember what is the basic makeup of many cell membranes, it is basically phospholipids. And what happens with phospholipase C is it will hydrolyze lecithin. Basically, it just pops off the hydrophilic phosphate heads. And hence, what it does is it just renders the phospholipids useless. It tears them apart and thereby destroys the cell membranes. Some strains of this particular bacteria will have also something called EXO-S, which stands for exoenzyme S, which prevents phagocytosis of bacteria and causes apoptosis of host cells. This bacteria, P. aeruginosa, is widespread in soil, water, fruits, and vegetables, can be introduced by dirt on shoes or contact lenses or contaminated cosmetics. The treatment and prevention. Prompt care of wounds, including the removal of dead tissue from burns, followed by the application of silver sulfdiazine cream. This is a topical. Bacteria resistant to many antibiotics. Some work, such as basically fluoroquinolones, gentamicin, and imipenem may work. For systemic infections, antibacterial drugs must be given intravenously and in high doses. Okay, so this is what we see there. Now what I'm going to do is I'm going to talk to you about diseases due to anaerobic bacterial wound infections. Now, wounds that create anaerobic conditions allow strict anaerobic species to colonize inside the tissue. So what you're going to see first is tetanus. Commonly, it was used to be called lockjaw. What you have for symptoms are restlessness, irritability, difficulty swallowing, muscle spasm, muscle pain in the jaw, abdomen, back, or entire body. What is the cause of this? Another Clostridium species called Clostridium tetani. Remember, this is going to be anaerobic. It is going to be spore forming. It is going to be a gram-positive rod. And what the bacteria do is they release a particular protein called tetanospasm, which blocks muscle relaxation. With tetanospasm, this is an exotoxin produced by the bacteria that act to block neurotransmitter release of inhibitory neurons, which would inhibit muscle contraction, thus maintaining neural contraction of the muscles. Okay? Here you have a child, an infant with neonatal tetanus, and you can see how it's arched back. Here is the spores forming in Clostridium tetani, okay? And you can see what's going on. They're rod-shaped, okay? Now, you see the endospores forming here. Here's the issue that you have to understand. With tetanus and the inhibitory neuron function, take a look at the arm. You have the normal flexion. The motor neuron stimulates, and what you're going to have is a muscle. The biceps contract. The triceps are going to relax. With normal extension, the triceps will contract, the biceps will relax. But with tetanus, both the triceps and the biceps contract, and the muscle, all of them are going to lock in, and the arm cannot move. Tetanus prevention and the management of wounds, basically what it is this. The crazy thing about this type of disease is that you have a vaccine that's available. You get reboosted after 10 years. Okay? And everybody's happy. Everybody's fine. If you talk to someone who just comes in an emergency room, it's unknown or fewer than 3 injections of the toxoid. If they've been fully vaccinated, 3 or more injections. If it's 5 years or less since the last dosage, they don't need any more vaccination. If it's 5 to 10 years since the last dose, they might, in certain more significant wounds, need a shot. If it's more than 10 years since the last dose, yes, they need the toxoid vaccination again, whether it's cleaning up minor wounds or much more profound wounds. Okay? If it's unknown, their history, or fewer than 3 injections, most of the time what's going to happen is they're going to recommend the injection, basically a shot. Okay? What you got to understand is that the organism is very common in the soils. The spores will contaminate wounds and germinate under anaerobic conditions, especially in dirty or puncture wounds. Okay? And what will also happen is that you're going to have a very sad situation with tetanus. And I say that's sad because it is common to see the incidence of tetanus show up as you have adults who are working on, let's say, manual jobs where they have the chance of getting minor wounds or major wounds. And if the vaccine is not available, particularly in developing countries, it's very common to get this disease. In the United States, I don't have the chart with me, but they will show that the incidence, which used to be much more when we were younger, because we have the Dtap vaccinations, which is basically diphtheria, pertussis, and of course the T part of it is tetanus. What happens is that people will not renew the tetanus after they get into young adulthood. So by about 30, 40, they're much more susceptible. And the risk of tetanus will increase as you start getting into the later 30s, 40, 50, 60. Because as these individuals are getting exposed, they don't think anything of it. And by the time that they're starting to have the onset of the disease, well, it may be too late, or they may have to get tetanus immune globulin. Okay? One of the important preventions is, of course, cleaning of the wound. Treatment with metrodiazonal and antitoxin can occur if someone does have it. That said, let us move to gas gangrene. This is clostridial myonecrosis. You want to keep in mind that clostridial, the vegetative growing cells, symptoms: severe pain, gas, fluid seeping from the wound, blackening of the overlying skin. Okay? Later, shock and death follow. Active agent, Clostridium perfringens. Sometimes other Clostridia species, but it's less likely. The presence of dirt and dead tissue in the wound accelerate the growth, as well as a long lag time to treat and clean the wound. The organism will grow in dead or poorly oxygenated tissue and release an alpha toxin. The alpha toxin diffuses from the injury, attacks lecithin, removes the phosphate head, thus destroying the cell membranes of tissues and leukocytes. Organisms grow in dead tissue medium, basically fermentation of amino acids and muscle glycogen. They will produce hydrogen and carbon dioxide, which accounts for the gaseous nature of gas gangrene. The mechanism of shock is not fully understood. It's very common with war wounds. impaired tissue circulation, as the patients that you see with diabetes or arteriosclerosis, or after self-induced abortions. Okay, prevention and treatment. Requires prompt cleaning and debridement of wounds. There is no vaccine that's available. Some value has been found with hyperbaric oxygen therapy. In other words, they put the person in a hyperbaric chamber and under enhanced pressure, let's say 3 atmospheres, you get a lot more oxygen available, and it will get much more deeper into different tissues and thereby inhibit the growth of this particular type of Clostridium. You have to keep in mind that penicillin will only act on the vegetative cells. The limits of antibiotics: they cannot stop bacterial growth or toxin production in large areas of dead tissue or in abscesses, since the diffusion is limited. And also, antibiotics will not inactivate the toxin. The next one we're going to deal with is actinomycosis, otherwise known as lumpy jaw. This can be a chronic disease. It is recurrent, painful swellings that open and drain pus. They heal with scarring, usually involving the face or neck. Some other sites will include chest, abdomen, or pelvis. The organism is Actinomyces israeli. Here is why they call it lumpy jaw. You'll see the inflammation there. And what you see there is this filamentous, gram-positive, slow-growing anaerobic bacteria. Usually it will start with a mouth wound. The organism is slow-growing, but extends to face, neck, and upper chest. In the tissue, the bacteria cells grow dense yellowish colonies called sulfur granules. Epidemiologically, there is no person-to-person spread. The organism is common in the mouth, intestine, and vaginal microbiota. Dental surgery, intestinal surgery, or insertion of IUDs can initiate the infections. There's been no proven measures to prevent it. They require prolonged treatment, sometimes weeks or months, due to the slow growth of the organism. Antibiotics include penicillin and tetracycline. And by the way, there is a very nice series of case presentation and microassessment that I encourage you to take time to review. The next area we're going to deal with is bacterial bite wound infections. Ah, yes, bites can be dangerous, even deadly. Look there, that's a Komodo Dragon. I know you're sitting there going, wait a minute, this is not a biology course, this is a microbiology course. Okay, here's one of the interesting things to know about this. The monitor lizard, otherwise known as the Komodo Dragon, has a very, very, very septic mouth. They have found 52 different pathogenic bacteria in their mouth. And you want to know something? You can see these very slow, mean, nasty lizards on their native islands. And what they literally will do is bite the prey. You know, it could be a water buffalo or something else. And they'll just sit by and wait for a bit. And then the organism that's been bitten gets overwhelmed with bacteremia, bloomp, and falls. And that's when you eat them. Now, the pathogens include Salmonella and Group A Streptococcus. And by the way, this is not just unusual for the monitor lizard. The African crocodile does the very same thing. Go up, give a good bite. The septic infection will occur, and then you can have lunch. And this is also how they started looking at, wait a minute, how come when these monitor lizards have fights, and they will have fights with other monitor lizards, how come they don't get these infections from their own dirty mouths? They found out that in their blood they have certain antimicrobial compounds that keep them from this, and they're presently looking at the development of these compounds for possibly antibiotic purposes. Well, let's first deal with human bites. Yeah. I refer you to table 23.9. Okay? And human bites are nasty. And that sounds like colloquialism, but here's the thing. You have a rapid onset pain, massive swelling, drainage of foul-smelling pus. You notice there's some really bad bites there, and they actually took away some of the tissue. You can see underneath the underlying tendons to control this. Human bite wounds. Okay, let's go through this. You're going to have a mixed mouth microbiota. This includes anaerobic streptococci, fusiform spirochetes, anaerobic gram-negative rods, sometimes Staphylococcus aureus. The combination of the bacteria works synergistically to destroy tissue. The crushing wounds from the bite enhance the anaerobic conditions of the wound. That's part of the problem there. Capsules of some species may evade phagocytosis, and if the tissue damage extends to tendons or other tissue, permanent loss of function can occur. Keep in mind that tendons are basically connective tissue. They are going to be cartilaginous, and I've taught this in A&P here. They're not heavily vascularized, which means that the transport of antibiotics into those tissues is going to be based on diffusion, so it's going to be slow, and the bacteria are going to have the better upper hand in causing tissue damage. Also, you've got the problem of the nature of the crushing wound will favor conditions for anaerobic bacteria growth. Synergistic infections. This is the sum effect of all organisms acting together is basically greater than the sum of their individual effects. The epidemiology. Usually this occurs with alcohol-related violence. Wouldn't you figure? Forcible restraint, or mouth care and the presence of dental disease. A lot of times, if you talk to law enforcement, usually the risk of bites is greater in alcohol-related violence, individuals that are on Skid Row that basically they've had alcohol, so they're not going to have much inhibition. Also forcible restraint, when you get into close combat fights, things like that, biting is going to occur. Okay? There is no proven preventative methods except to avoid altercations. The proper cleaning and disinfecting of wounds may help. The treatment may actually require surgery to remove the crushed and damaged tissue that's becoming the culture area for a lot of these anaerobic organisms. Next is Pasteurella multisida bite wound infections. Okay? What you have for symptoms are the spreading redness, tenderness, swelling, and discharge of pus. Pasteurella multisida is a gram-negative, facultatively anaerobic, encapsulated coccobacillus. This is introduced by a bite. The bacteria resist phagocytosis due to the capsule. They may release cell-destroying toxins, and abscesses will form. They can be opsonized by a specific antibody, then phagocytized and killed. If untreated, they can cause bacteremia, resulting in endocarditis or meningitis. Epidemiology. This is referred to as fowl cholera. Pasteur first discovered the pathogen and its effects on chicken. Bacteria are found in various animals. Cat bites may have it more than dogs. The treatment is usually penicillin or augmentin, and the augmentin is favored in some cases because of the beta-lactamase inhibitor. Immediate cleaning and quick medical attention may help prevent the infection. There is no vaccine. Then we come up to cat scratch disease, Bartonellosis. Bartonellosis starts, a pimple will appear at the bite or scratch site, followed by localized lymph node enlargement, fever. The nodes may soften and actually drain pus. If you have a prolonged fever, you're getting worse and worse, and then convulsions, and this is an indicative of other body parts being involved. Bartonella henselae is a curved gram-negative rod. The infection is not completely understood, but the bacteria will spread via the lymph system. It's usually halted in the lymph system, but if it spreads to the bloodstream, the bacteria will infect the heart, brain, and other organs. The complications are most often seen with immunosuppressed patients, such as AIDS patients. Pilosis hepatitis. This is blood-filled cysts that form in the liver. Bacterial angiomatosis are nodules composed of proliferating blood vessels which develop in the skin and other parts of the body. The bacteria is present in those lesions. Epidemiologically, this is a zoonosis of cats. It's spread by cat fleas. The fleas have the bacteria, and they will spread it in flea bites or actually in flea feces. Infected cats many times are asymptomatic. The humans are the accidental hosts, and mainly the disease occurs in those under the age of 18. Prevention and treatment: avoid rough play with cats. Promptly clean and disinfect skin breaks. Flea control of the cats. Most of these bacteria can be treated with ampicillin. Next, streptobacillary rat bite fever. This is limited in its exposure, usually poor people in large cities where there's presence of rats, such as in slums, etc. Also, laboratory rat handlers will get this. The symptoms are chills, fever, muscle aches, headaches, and vomiting. Later, there's a rash and pain in one or more of the large joints. The causative agent is Streptobacillus Moniliformis, which is a pleomorphic gram-negative bacteria. Spontaneously produces L forms, and L forms are cell wall deficient bacteria variants. They resemble mycoplasmas. They're usually avirulent. The pathology is a bite wound without treatment. The bacteria quickly invade the bloodstream, with fevers appearing and subsiding. A lot of times the patient will recover without treatment, but if the infection spreads to various body organs, death may occur. Epidemiology: wild and lab rats can carry this bacteria. The animals that prey on these infected rats can also carry the bacteria to humans. Food. This is referred to as Haverhill fever, or water contaminated by rat feces can also transmit the infection. Prevention and treatment: control of wild rats and mice, proper lab care of lab rats. The treatment, if you have an infection, is penicillin. Okay, now we go into fungal wound infections as we wind down this. A couple of things to keep in mind. As always, I add on a little bit beyond the chapter, so you need to follow with the notes here. The fungal wound infections, what you got to understand is that fungal infections of wounds are more serious than dermal mycosis. Typically, soil fungi enter through the injuries that permit entry into the subcutaneous tissue. And once you're in there, then it's a completely different ballgame. Sporotrichosis, otherwise known as Rose Gardner's disease. And you can see the image there. You also see, notice how the pattern goes through basically the lymph system. Here's another image of it you'll see in your book, figure 2314. Okay, now what you have is a painless ulcerating nodules appearing that follow the lymphatic system, usually away from the initial injury site, and may travel towards the center of the body. Now, the cause of this is Sporothrix schneckii, and this is a dimorphic fungi, so you're going to see both a mold form and a yeast form. Remember, the yeast form will be sort of the rounded somewhat cells, and you see this in the lower image. The upper form, you see actually hyphae and conidia, so that is the mold form. The spores will multiply at the site of the introduction, usually, for example, a thorn, a sliver, a splinter, or other plant material, and that causes a small nodule that ulcerates. The spores are then carried by lymph flow, and they repeat ulceration process in the course of the lymphatic vessel. The ulceration and spore transport may occur beneath the skin regardless of the absence of lymphatic vessels. Epidemiologically, there is a worldwide presence of these spores. Occupations with sharp plant materials increase the risk of infection. So what you have to think about, it is not just rose gardeners. Anybody that deals with plants, let's say, raspberry pickers, individuals that pick raspberries that have thorns, or they deal with sea thorn plants where they pick the fruit. All of this is going to be a possibility for a risk of infection. Usually the prevention and treatment, protective gloves and clothing. Most cases can be treated, interestingly enough, with just a potassium iodide, which somehow enhances the body's ability to fight off the fungus. Okay? Generalized infections require amphotericin B and icotrionazole. The next one is known as Medora Foot, also known as Medora mycosis or eumycetoma. This is basically a hardened swelling of the foot, pus-filled sinus tracts in the foot filled with fungal grains. Inoculation is basically into a scratch or abrasion any of a number of fungi, penicillium, aspergillus, magirella, actinomyces, such as pneumocardia. Now, Pseudomonas Aeruginosa is the most common etiological agent of a eumycetoma in the United States. Okay, a lot of times this is due to, it was first reported in the region of Medora in southern India. Okay, basically, the individuals walk all the time without any foot covering. Okay, the fungal infection of the subcutaneous and the cutaneous tissue occurs. The infection destroys the soft tissue and nearby related tissue. This can actually go into the bone tissue. Okay. As I said, the disease was first reported in the region of Medora in southern India. Fungal infections are very similar, but may be caused by local fungi in different parts of the world. The best way to treat this is with dapsone and tetracyclines. Well, now you've completed Lecture 13. Congratulations. I do encourage you to review the microassessment, the diseases in review, the future opportunities, and this chapter summary for Chapter 23. And this concludes our lecture, and I hope you have a nice day.